So now we're going to turn it over to Krista Jackson. Krista joined the lab in March of last year. She entered the program like many of you, expecting to do medical school next. She expected to do the one-year program. And then made the possibly ill-advised decision to continue the second year, even though she had been accepted to medical school, and defer for a year so that she can complete her master's and then be in a stronger position for future residency slots. So I will now turn this over to Krista.
Okay, how about now? Better? Yeah. Sweet. Okay, so curing osteoporosis before it kills me, because if you remember from your physiology lecture, I am a small white female, so I'm basically doomed. Like, it's coming for me, and we're trying to get to the cure before I get there. So before I even join the lab, and before I even get to joining the lab, we will back up and cover osteoporosis in case you've forgotten that and you're crammed for finals. Simplest definition, osteoporosis is bad. A more detailed definition, osteoporosis is a skeletal disorder characterized by compromised bone strength, predisposing a person to an increased risk for fracture. On a more operational level, osteoporosis is when bone resorption exceeds bone formation, because hopefully you guys remember osteoblast, osteoclast, there's that dynamic relationship between them, and osteoporosis is when you have more resorption from the osteoclast than formation from the osteoblast. So, before I joined the lab, back in 2015, Dr. Lowery published a paper indicating that when you knock out the BMPR2 gene in mice, those mice have increased bone density. It was an incidental finding. They weren't expecting it, but when they found it, they were like, okay, this is really cool. Let's see what happens here. So what they find is, if you look at this chart, the black line is wild-type mice, the red line is mutant mice, which are the mice that had the BMPR2 knockout, and the gray bars on either side are the confidence intervals. So the mice that had the BMPR2 knockout had roughly a 1.5-fold increase in bone density at 15 weeks of age. And then as those mice age, like, you're always going to decrease in bone density as you age. It's just going to happen. But as they age, they were partially protected from that age-related decline. So when you get out to 55 weeks, which is, pretty good age for a mouse, you have almost a 4-fold increase in the bone density, which is huge, because if we could replicate that in humans, I mean, I'm saved, probably. So they decided to try to figure out, okay, like, what is causing that increase in bone density? And to do that, they looked at serum marker levels. Now, P1NP is a serum marker for osteoblast function, which is, I remember, is bone formation. And CTX is a marker for osteoclast activity, which is bone resorption. So I only have the P1NP levels up here because CTX levels were relatively unchanged at 35 weeks and 55 weeks. So it's fairly boring. And basically all that tells us is that osteoclast activity isn't really changing. But the P1NP levels, which are more exciting, at 35 weeks, you've got, again, the red dots are the mutant mice and the black dots are the wild-type mice. And you can see that there's a significant increase in P1NP levels, but that's returned to practically normal at 55 weeks. So clearly what's driving this increased bone density is osteoblast activity without a change in osteoclast activity. So that gets us to our hypothesis. The significance of everything that I've just told you is that we think that these BMPR2 knockout mice can serve as a model for identifying novel regulators of osteoblast activity in the aging skeleton, and that could reveal novel treatment strategies. So my hypothesis is that if we do whole transcriptome profiling of BMPR2 knockout mice, we can identify a gene signature associated with that increased osteoblast activity in the aging skeleton. To that end, we decided to try to find that gene signature, whatever is driving the increased osteoblast activity. So we took wild-type mice and we took mutant mice and we took their bones, because they are dead now, rest in peace. And we fleshed out the bone marrow. So here in the picture, we've got over here on the left, that's the bones with the bone marrow in them. And on the right, you can barely see it, but we fleshed out all the bone marrow so that we wouldn't have any contamination. We just wanted transcripts from the bones themselves. And we sent them off to a company called GeneWiz, and they did all of the RNA sequencing analysis. And then they sent us back massive amounts of data that made me want to cry. So my job then was to filter through all of that data and try to find the smoking gun, as it were, the genes that are causing this increase in osteoblast activity. So I filtered the genes a few different ways. I first filtered to only exons. I didn't want any introns because I didn't want to risk any sort of contamination. I just wanted the bone transcripts. I also filtered out all the pseudogenes. There were tons of pseudogenes with these little fragments of genes that are just murking up my data and not actually transcribing for anything. We then use housekeeping genes. A lot of people, when they filter with housekeeping genes, they'll use 1 or 2, maybe 3 or 4 housekeeping genes. We wanted to be really stringent, so we used 8 housekeeping genes. And I'm not sure how familiar you guys are with housekeeping genes. They're genes that are widely accepted to be relatively stable levels throughout a mouse's age, throughout a mouse as they age. So we compared all of the genes to these housekeeping genes. Anything that had a 2-fold or greater increase in expression level compared to the housekeepers, we said, all right, that is an upregulated gene. Anything that had a 2-fold or less decrease, we said, all right, those are downregulated compared to the housekeeping genes. And anything that fell in between 2-fold or up or down, we said, all right, those are relatively normal levels of expression in those genes. So I did all of that for all of the data for the mice at 35 weeks of age, because if you remember, 35 weeks is when we had that increased osteoblast activity, which is what we're really excited about. Unfortunately, I still had a massive amount of data and no smoking gun, and I'd run out of ways to filter my 35-week-old genes. So, fortunately, we also had a snapshot of the genes at 55 weeks, because if you remember from the picture, 55 weeks is when everything's returned back to relatively normal. So whatever was driving things at 35 weeks has kind of slacked off at 55 weeks. So we took the 55-week data and we did the exact same thing, figured out who's upregulated, who's downregulated, filtered out the pseudogenes, and then we compared everybody to each other. Genes at 35 weeks, how do they look compared to genes at 55 weeks? Because whatever's going on, there's got to be a difference there. Something has changed. And for that, we created the bane of my existence, a 15-zone Venn diagram with a very beautiful legend that took me weeks to put together. So just for example, like zone 1, which is right here in the center, you've got almost 16,000 genes. Those are all the genes that are normal levels at 35 weeks and then also normal levels at 55 weeks, which is great. There's a ton of them because they're not interesting to me and I can filter all of that stuff out. I don't even care about those. But zone 2 over here, we've got all the genes that are upregulated at 35 weeks, and their levels are so low they're not even detected by RNA sequencing at 55 weeks. So that's really interesting because that could be what's driving it, and there's only 9 genes, which really narrows it down. The problem is we don't know if this is a driver, something that's like overexpressed at 35 weeks and not really expressed at 55 weeks. We don't know if it could be a repressor gene, something that is downregulated at 35 weeks, so the osteoblasts are going crazy, and then it picks back up at 55 weeks, so they calm back down. We have no idea, so there's tons of options, and I haven't been able to eliminate anything yet, which is why I still have to include all of these zones and occasionally want to cry. So the next way we filtered them was through the enricher analysis, because even though I'd done all of this data sorting everything, we still didn't even know if we were on the right track. But there's this program called Enricher that does bioinformatics analysis, and we ran all of the different zones in the Venn diagram through those. And bioinformatics, there's all sorts of things, the transcription, pathways, ontologies, blah, blah, blah. The bottom line is we ran all the zones separately, and then we ran certain zones that we picked out as like strong putative drivers or strong putative repressors, things that could be things that we thought had potential. And what we found was, I've included some data from zones 2 and 15, which we identified as strong putative drivers. And they are highly enriched for transcription factor binding sites in known skeletal pathways, which basically just tells us, okay, we're on the right track. We've got SMAT1, we've got FOXP1. We're getting closer. We haven't just completely gone down a rabbit hole. So our results on our future directions, as Lowery said in a meeting we had this week, we haven't found the gun, but we can see the smoke. And I actually took this picture of him without him knowing while we were trying to map out all of the different options. Because like I said, we don't know if it's a driver, we don't know if it's a repressor, we don't know if it's a hybrid option between drivers and repressors. So we were literally just sitting in front of a whiteboard, mapping out all of the different possibilities and then crossing them out. And we're like, okay, this is an option. No, that doesn't work. Move on. So we're doing a lot of that. We're trying to narrow down that gene signature even further. We're also working with antibody arrays to back up our data. We really want to obtain some knockout mice. Once we find genes that we think are really particularly interesting, we want to con some people into giving us some of their mice and see if we can check the bone density levels in those mice, depending on which we think are repressors or drivers. And we're probably going to continue to bang our heads occasionally. So for my acknowledgements, I'd like to thank Dr. Jonathan Lowery for taking a chance on me. I had absolutely no research experience coming to him, and he still took me into his lab and has taught me everything. I'd like to thank Aaron Hudnall, who has done previous work on this research and is actually the student that introduced me to Dr. Lowery. Nick, who has done absolutely nothing for my project, but he deals with my regular insanity in the lab when he's not, sleeping at his lab bench. The BMRG, which is on Twitter, you should follow them. They're very funny. Occasionally they let me tweet for them. And the Marian University Faculty Research Development Grant and the Indiana Academy of Science Senior Research Grant for funding what we do. So if you have any questions, I would love to answer.